recording. And now I'll share it again. We don't need this. So let's talk about where we're at. So we're doing Lab 4 on stains today. I mentioned something in the lecture about the lab, and I'm trying to remember what the heck I said. Does anyone remember what I said about the lab? I think it was that the lab reports, not lab reports, the lab worksheets are due 11:59 p.m. on Saturday. And some of you have been turning them in late because I always make corrections and then tell you try again or something. You have a few days to update it. The last one, I finished it really late on Sunday. It might have been Monday morning for a few people. And so I decided I was going to give you a little bit more time to do an update. And so they're now due for the previous two labs, I think. They're due on Thursday, 11:59 p.m. on Thursday. So take a look at that. If you didn't get 100%, you can resubmit it. Those of you who did not resubmit it, I may grade you as late because it is late. And I might not dock you off 50%, but it's realized I want those in 11:59 p.m. on Saturday. And in the future, meaning for labs 2 and 3, I won't dock you 50%. But in the future, all labs, if you turn them in after Saturday, meaning after 11:59 p.m. on Saturday, I'm just going to grade it as it's turned in late. Okay? Meaning it's supposed to be in 11:59 p.m. on Saturday. All right, any questions about what we're doing or anything I said? If not, let's get on to the lab. Lab 4, staining techniques. We'll talk about differential, simple, and special stains. There's a number of reading assignments for this lab. Sorry here, I'm about to sneeze and I don't know why. You need to read, I'm just going to say about these different things here. It's a fair amount when you all put it together, but each one is just a few pages. They're all covering a stain that we're going to talk about in this lab. You will be working on some video clips and some virtual labs. Let me state that the virtual labs, I do want you to do them, but if they are Flash, a virtual lab made of Flash, then Flash is no longer supported and you may have trouble getting it to run. Usually I have to just double click, meaning it'll tell me Flash is no longer supported and we don't recommend watching it or whatever. And I double click on it again and then I can get it to play. And hopefully you guys can do the same. But if you can't get Flash to run, then email me and I'll see what we can do. Hopefully the virtual labs will run for you. For your learning objectives, you should be able to know how to prepare a bacterial smear, and you make a bacterial smear before you do the staining. So whenever you do the staining, you first, at least most of the time when you do staining, I'm not going to say all the time, but most of the time when you do a staining, you have to make a bacterial smear. Understand the principles and the procedures used when performing a stain, such as a simple stain, a differential stain, and a special stain. Be able to interpret the results for each of these stains, a simple stain, a differential stain, and a special stain. Be able to define the following terms: what's a simple stain, a differential stain, a special stain, a coccus, bacillus, spirillum, peptidoglycan. What's a gram-positive cell, a gram-negative cell, acid-fast cell or an acid-fast staining cell, a primary stain, a mordant, a decolorizer, a counterstain. What's a capsule, an endospore, and a flagella. There was something I was going to talk about. A simple stain is very easy to define. A differential stain is fairly easy to define. A special stain really is a stain that's done for special purposes, to stain special things inside the cell, and so it's not so easy to define exactly what a special stain is, but one easy way to discuss it is it's a stain that's not a simple stain, and it's not a differential stain. It's what's left. Okay, we're using a special stain to specifically stain special structures of a cell. All right, any questions about any of that? Hopefully I haven't confused you on a special stain. Because of their extremely small size, bacteria are essentially transparent when you're trying to view them under the microscope. Therefore, it's necessary to stain them before you observe them. And before you can observe them and stain them, you have to prepare a bacterial smear. You add a drop of sterile water on the clean slide, and then you add the bacteria and you mix it in and then spread that water all over the slide. You then air dry the slide and then heat fix it. The reason why you do those two steps is you need to do something to attach the cells to the glass slide. If you don't air dry and heat fix the cells onto the slide, then they will wash off in the staining procedure, especially with your water wash of the staining procedure. Any question about any of that? So that's what a bacterial smear is. Done before you do the stain. A simple stain is just where you apply one stain to the bacteria cells. And it says you're applying one basic stain. In reality, it could be one negative stain. It's just one stain. Most of the time it is a basic stain. And if you just stain the cells with crystal violet or methylene blue, those are the two most commonly used stains, that is a simple stain, because it's only using one stain. A differential stain uses two or more stains or dyes, and it provides additional information about the cell. So a simple stain uses one stain and all the cells are colored the same. Like if you stain with crystal violet, all the cells will stain purple. But if you do a differential stain, some of the cells will stain one color and the other cells will stain another color. And that's why we call it differential. It allows you to determine differences between cells. Any question about that? Sorry, I'm trying to keep myself from sneezing, and that I was hoping would help. It's not exactly help, but it helped a little. The gram stain is the most commonly used differential stain, allowing you to differentiate between gram-positive cells and gram-negative cells. And we'll talk about exactly what that is in this lab. Lastly, there are special stains, and they're used to stain a particular structure in a cell. You can use a special stain to stain the capsules, to stain endospores, and to stain flagella. And there are special stains, actually there's more than one, but there's at least one to stain a capsule, and another one to stain an endospore, and a third one to stain a flagella. Any question about that? You should know about the common bacteria shapes. Bacteria can be cocci. That can be pronounced in different ways as cocci, cocky, and cockeye. Did I say 4 different ways? I think I only said 3. Sorry. I don't remember what the 4th one is. And I don't remember what I even said, so I'm not going to even try and get it. But all of those are correct. Different parts of the world use different names. Different parts of the United States use different names. I don't care how you pronounce it. The second most, well, actually this is one of the most common cells. The other second most common type of cell is a bacilli, and you can also call that a rod. They're rod-shaped shells. The cocci are balls. The third type of a cell is a spirilla, a spiral, and this is showing you spirilla. And don't ask me why they're calling it in this picture a spirilla bacilli, because that is a spirilla, not a bacilli. Any questions about that? So call this a spirilla and this a bacilli. Do not call these bacilli. Do not call these spirilla. Those are the 3 most common shapes. There are some other shapes. We're not going to talk about any of them except for a coccobacilli, and that is if you shorten the bacilli and get them very, very short, you have a coccobacilli, and they're in between a cocci and a bacilli. Any question about that? You should also know the cellular growth arrangement. That is how, when the cells are growing, how they stick together. So you can have singlets, and actually the spirilla only have singlets, but there can be singlets of cocci or singlets of bacilli. There can also be diplococci, where the cocci stick together as 2 cocci together, diplococci. Anyone know a diplococci? There are also cocci. Some species can grow as tetrads. That's 4 cells stuck together, and generally only the cocci are tetrads. You're going to have Sarcsinia. That's where the cocci as a tetrad divides into 8 cells, so there will be 8 stuck together. And then there can be streptococci. That's where they form a chain. And then lastly, the cocci can be staphylococci, and this means looks like a grape cluster, and that's where the cocci grow in a cluster like a cluster of grapes. Only the cocci form staphylococci and tetrads, and you don't need to know about Sarcsinia for this lab, but only the cocci form tetrads and Sarcsinia. The bacilli can be streptobacilli, so that they can form together like a, what do you call that, a sausage link, one after the other. And there can be diplobacilli. Let me see if I can show you one more type of bacilli that's not up here. I'm not sure I'll be able to do it here because I got to draw, and I'm not good at drawing from this. See if I can do it with a smart art. Nope. Let me try again. I don't know how to get paint from here if it isn't there. Um, I'll try and demonstrate with pencils maybe. That'll be good. So the bacilli in a chain can form sausage links. And this is sort of a sausage link. Hopefully you guys can see that. Let me get it further away. There. So there's 3 of them now, because I'm using the cap as one. All right? So that's streptobacilli. But you can have the bacilli growing instead of end to end this way, they can be growing this way. Get it a little closer so you can see it. Can you see that? And this is called a palisade when they're growing that way. All right, any question about any of that? Anything else I need to talk about here? No. When you're talking about how they're growing, it's called a cellular growth arrangement or a growth arrangement. And the shapes are just the individual cells. So the growth arrangement is when you see a combination of cells stuck together. And we'll talk a little bit more about that in a later lab. To get the cellular growth arrangement, you have to grow the cells in a liquid culture, because if they don't grow in a liquid culture, they can stick together. Like when you grow it on a petri dish, a solid culture, they can stick together, and then trying to determine the growth arrangement is difficult, if not impossible, to do. You need to get the growth arrangement, you need to grow it in a liquid culture, and then the cells can be free to form their cellular growth arrangement. Any question about any of that?
Question.
Yeah.
Can these bacteria, be mingled together, and how they will be named?
I'm not understanding your question. Can you repeat it?
All right. Like, for example, can these bacteria, join together, and how they will be called?
How they form these different growth arrangements is just how the cells stick together when the cell is dividing.
Okay.
Different species stick together in different arrangements. Okay? And I didn't want to throw in a complication, but there is one complication, and that is you can have more than one growth arrangement, and that's because like a streptococci, well, it can break. And when it breaks, like this one, or if it were to break, we would have streptococci, this one that's not broken, and then this one would be diplococci, and then one of the diplococci could break, and then you got a singlet. So usually you will have more than one growth arrangement in the cells. And what you do is you call it by the highest growth arrangement, the streptococci. Okay? And I didn't really want to talk about that here, but that is true. Meaning you're not going to see one growth arrangement.
I ask because, I'm doing research, I'm part of a biomedical research team, so that's why.
You'll see more than one growth arrangement, but if you want to see growth arrangement, let me emphasize, you have to grow the bacteria in a liquid culture called the broth. Okay? All right, let's move on. So let's talk about differential stains. The most important is the Gram stain. It was invented in 1884. You don't need to know that, but you should know it was invented by Hans Christian Gram, and that's why the Gram stain should always be used a capital G, because it's named after somebody. It is a differential stain, allowing you to separate the cells into 2 types, The gram-positive cells, they retain the crystal violet and stain purple, and the gram-negative cells, and they do not retain the first stain, the crystal violet, and they stain by the second stain reddish or pinkish. And these we call gram-negative. Whoops, didn't mean to change that. Move that back. All right, any question about any of that? So here we're looking at different cells and how they stain. These right here, these rods are purple, so they're gram positive. That's a purple cockeye right in this region. Cockeye. Coccus, really. Here's a purple rod or bacillus, so that's gram negative. I mean, not a purple, it's a pinkish purple, but a pinkish gram negative bacillus. That looks like a spirilla, but it could be two curved rods next to each other. And there's a spirilla, gram negative spirilla. All right. Gram reactions are based on the ability of the cell wall to retain the crystal violet. When we look at bacteria, there are two basic shapes for the bacteria. There are the gram-positive microorganisms that have a thick peptidoglycan layer in their cell wall. That's the cell membrane, the plasma membrane, and this is the cell wall. The gram-positive cells have a very thick peptidoglycan layer, and the crystal violet gets stuck in the peptidoglycan, and it doesn't wash off in the gram stain. The gram-negative cells, they do typically have peptidoglycan, but it's a much thinner layer. Note that it's more than one layer, because this is, well, we'll look at that later, but it's layer upon layer of peptidoglycan, but the layers are much thinner in the gram-negative cells than in the gram-positive cells. The gram-negative cells also have lipids on the outer portion of their cell wall, and we'll talk more about that later. And in the gram-negative cells, this thin layer of peptidoglycan isn't enough to retain the crystal violet, so it washes out of the cells in the gram stain. Any question about that? So the gram stain is literally showing you what the cell wall looks like. If it's gram positive, it looks like this. If it's gram negative, the cell wall looks like this. Typically. There are some exceptions. We'll talk about one of those exceptions in this lab. All right. So you should know what a mordant is. I already discussed in the lecture, so I'm not going to talk about it here. Some gram-positive bacteria lose their ability to retain crystal violet and are easily discolorized. Therefore, they're called gram variable, because when you do a gram stain on this species, you can see that some cells are staining gram positive and other cells are staining gram negatively. This usually happens in cells in the family of Mycobacterium because these cells have a lot of macolic acid in their cell wall, and the macolic acid resists the stains of the gram stain from getting in the cell, so that the stain does not stain the cell well. Sometimes, if the Mycobacterium are very resistant to the stains of the gram stain, meaning the crystal violet and the safranin, the cells won't stain. Most of the time they do stain lightly. Okay? How you can avoid the gram variable results is always use newly grown cells. What can you notice about the gram-positive cells here compared to the gram-negative cells?
They're very dark.
Yeah, they're very dark. What else? What do you notice about that cell there in particular compared to, let's say, that cell or that cell? And then there's this
It's
cell.
got a little bit more shape to it.
Yeah, it's very distinct. The gram-positive staining cells are very sharp and distinct. And the gram-negative cells, especially some of them, like that one there, it's very faint and it seems like it's disappearing. It's because these cells are dying, or that one's probably dead, and the cell is disintegrating. That one there too. And the dead cells, they're dying or dead, so they're not retaining the crystal violet, and that's understandable. So what you want to do is stain cells that have been freshly grown to do your gram stain, and then you won't have gram variable results. Old cells, meaning older than 2 days old, do not stain well in the gram stain. The gram stain requires freshly grown cells. And these cells, the very sharp ones, they're freshly grown or fresh new cells. Okay? And then these cells, they're not new and they're not staining well. So that's one thing you can do to remove gram variable. Whenever I've had students who had a gram variable result, I ask them, how old are your cells? And most of the time they would tell me, oh, they're about 2 weeks old. And I said, well, that's why it's not staining well. Gram stain has to be done on freshly grown cells. Cells that are 2 days or younger. All right, any question about any of that? And then there's some tips there. Avoid using old cells. You should also have a prepare a thin smear. If you make your bacterial smear very thick, which is kind of hard to explain. I don't know if you can hear that. There's some banging going on outside my window. Somebody might be lighting off firecrackers. If you have a preparation of cell that's very thick, it will be difficult to get the stain into all of the cells, and then also it'll be difficult to get the stain to wash out if it's supposed to wash out. So you want a very thin layer of cells. When you make a bacterial smear, the ideal is to have one layer of cells. Okay? Any question about that? Meaning you don't use very many cells and you tend to spread them out. So prepare thin smears. Heat fix the bacteria. Well, if you don't heat fix and air dry, the cells are going to wash off. I'm not sure why that'll help with the gram variable. And avoid overheating the cells during heat fixation. If you overheat the cells, you will get the cells to melt and the cell wall will be damaged. You should also use fresh staining reagents. Don't over decolorize or under decolorize. All right, so those are some tips to prevent the gram variable. And the main one is use fresh cells. I've only seen gram variable results with old cells. Oops, sorry. All right, any questions about the gram stain? If not, let's talk about our next stain. The second most common differential stain is an acid-fast stain. Some bacteria, such as Mycobacterium and Nocardia, have a waxy substance called mycolic acid in their cell walls. And these bacteria resist the decolorization of an acid stain. So in this stain you have an acid alcohol decolorization step, and these cells resist the removal of the acid-fast stain. And we call these acid-fast bacteria. Mycobacterium pneumonia is an example of an acid-fast staining bacteria. You can read about that stain. Here's an acid-fast stain, and the acid-fast bacteria stain pinkish. Oh, there it is there. Usually they're rods, the Mycobacterium, and they tend to stick together so they're hard to see, but that's why it's pinkish there and pinkish there and pinkish there. The acid-fast negative bacteria, meaning the acid-fast negative bacteria, stain the second dye you use. In this case it's green, but it could also be blue. There are also special stains, and these are stains to determine if a cell has special structures which the stain stains. One special stain is staining for the glycocalyx, also pronounced glycocalyx. If the glycocalyx is firmly attached to the cell wall, and if you can see it under the light microscope, it is referred to as a capsule, shown here. The glycocalyx is something external to the cell wall that the cell secretes, and so the capsule, or the glycocalyx, is something outside the cell. A structure the cell makes, but it's outside the cell. Any question about that? And you can use a negative stain, a special stain, to stain for a capsule. Capsules are important because the production of a capsule can be associated with the bacteria's virulence, meaning how pathogenic the bacteria is. Capsules usually look very mucid, meaning wet and mucousy, like when you grow them in a culture. And this is Streptococcus pneumonia, which forms a capsule and looks very mucid on the colonies on the plate. Here's a capsule stain, staining for Klebsiella pneumonia. And it says using India ink, but this is not India ink, because India ink stains the background and it lightly stains the cells. And that would be a negative stain and a capsule stain using India ink. This picture I know is not India ink, and I'll show you a real picture of India ink. It's similar to India ink. The point is that the capsule stain shows you the cell, and then there's a region around the cell, which is called the capsule, which is not staining, and then the background is stained, at least with a negative stain, and that allows you to see the capsule. Any question about any of that? All right. A special stain can also be an endospore stain, and be careful on this one because some endospore stains are a special stain, and some endospore stains are a differential stain. Okay, I know that's confusing. In this lab here, we're using an endospore stain that is a special stain. Did I confuse anybody? All right. Endospores are metabolically fairly inactive cells. They're hardy structures where the cells are in stasis, sort of like hibernating, waiting for the environment to become favorable for the cells to grow. And endospores are produced by some bacteria as a defensive strategy for surviving in an unfavorable environment. When the environment becomes favorable once again, the endospore will germinate, and then the cells will come out of the endospore, germinating out of the endospore, and grow and become metabolically active. And those cells are their normal metabolic state, and that is a vegetative state. A vegetative state is the normal cell growing and metabolically active. In endospore, the cells are essentially metabolically inactive, and they're just surviving, waiting for the environment to become more favorable. This table down here lists some of the pathogenic endospore forming bacilli. Largely, the endospores are two families, Bacillus and Clostridium. And these bacteria are important because they do cause a number of human diseases. So these are pathogenic bacteria because they cause human diseases. And you'll need to use this table to answer some of the questions in the worksheet. And here's a stain for staining for endospores. The endospore is a structure the cell makes. Let me see if I can blow this up. So it's made inside the cell. Here's a cell here, and the red parts are the vegetative cell, and the green part is the endospore inside the cell. And there's another one. The cell's right there, and there's the endospore, and there's the rest of the cell. And there's another one. You can see endospores inside the cell, like that one, and here's another one right there. It's a real good one. And then sometimes you can see the endospore that's already left the cell. The cell, when it dies, it will release the endospore. And this right there is a free endospore, so is that right there. And this stain here, the endospore stain green from malachite green, that's a stain. And the vegetative cell stain pinkish. At least in this stain here. Any question about the endospore stain? I'll move this over here and talk about the next cells. Another special stain is looking for flagella. Flagella are long thread-like, kind of like a whip appendage that cells have. And if the bacteria cell can swim or move in a liquid, then it is moving because it has flagella. Bacteria, the only way it can swim is if it has flagella. And there's a special stain for looking to see if the cell forms flagella for purposes of motility. Normally when you stain the cells, the flagella are so thin that they do not show up. And you need a special stain in order for the flagella to show up. You do need to know this, I don't know, you want to call it figure or table or image here of the thing for answering one of the questions, but I'm not going to test you on what a monotrichous flagella flagellated arrangement is. I'm not going to test you on that, but you do need to answer the question. And when you answer the question, it's going to show you this picture again. There are some cells which have flagella. These are all one species. I know that looks hard to believe because they look different, but this is a Coccobacillus. Some of them are small like that, and then some of the Coccobacillus are actually very large and shaped like bacilli. There's just some species of Coccobacillus that they are pleomorphic in their shape, and this is one of them. When you look at the flagella of one species, you base it on the highest number. And so this would be a well, I shouldn't tell you what the thing is. Sorry. Peritrichous flagellated arrangement. Some of the people, like this cell here, it looks like it has 2 or 3 from one end, so that would be Lophotrichus. But this is the same species as that one and this one, and so we go it on this one. Is that clear to everyone? You call it by the highest number. And I think the reason why this one only has 3 is it's making the flagella, and in fact there could even be 1 right there, very faintly seen right there. All right, any question about any of that? I already mentioned you need a special stain to show flagella. What the special stain does is that it coats the flagella one time, and then another time, and then another time, and then another time, so that there's so many coatings put on the flagella that you can now see them under the light microscope. Any questions about any of that? All right, know the terms of this lab. You don't need to know the references, but then do the laboratory 4 exercises. You're not physically going to be performing any of these activities, but you must understand how to prepare bacterial smears for staining. Procedure 1 covers preparing bacterial smears, and we have some video clips to show you how to do that. Preparing the bacterial smear, then the air drying, I don't know if that's in here or here, and then the heat fixing. And then we're doing procedure 2, a simple stain. A video clip, I'm doing a simple stain, and then we're looking at a simple stain here. All of the cells stain the same color with the simple stain. It's using only 1 dye, so of course they're staining. And then answer the questions for the procedures. Procedure 3, we're going through the Gram stain. We have a video on the Gram stain and a virtual lab. Try and go through the virtual lab and then answer the questions on the Gram stain. I think you have to fill that table in. And then predict the color you will see from the bacteria. You may need to look up, is Staphylococcus aureus gram positive or gram negative? And I don't know if it's mentioned someplace in this lab, or you have to look it up, and then give the color you expect to see. Pink, purple, or colorless after the primary stain is added. Okay? And all the answers would be pink, purple, or colorless. Procedure 4, you're looking at the acid-fast stain, where the acid-fast bacteria will stain acid-fast positive, and then the acid-fast negative bacteria don't pick up the stain. The acid-fast cells stain pinkish, and at the end they'll remain pinkish, and then the acid-fast negative cells in this procedure stain green. We have a little video discussing that. And then here you're looking at a preparation of cells, and then answer the questions. Procedure 5, you're looking at special stains, and there's 3 special stains I can tell by looking at them. You're looking at obviously the endospore staining, a negative stain, and the flagella stain. And then answer the questions. And then there's case studies. Read the case study, try and figure out what's going on, take a look at the results. Here we see an X-ray indicating some lung condensation. They're then looking at the sputum smear. That's something you spit out of your mouth. And then the stain of the sputum showing two different colored cells, greenish and pinkish. And then answer the questions of the case study. Let me state, when it's asking for the most probable diagnosis, you can state the disease if you specifically state one disease, but it'd be easier to give the species that's causing the infection. Okay? So if you don't give a general disease, I mean, give it the specific disease, it would be wrong. Like if you were to say something like pneumonia. Well, there's many different species that can cause pneumonia. That may not be the answer for this, by the way. So you have to give what is causing the pneumonia. Or you could say maybe streptococcus, pneumonia, something like that, meaning the disease caused by a specific organism. In case study 2, you have another case study, and it's asking you what is causing this disease of the patient. And it's giving you some information, and there's the Gram stain, so you know it's gram-positive infection. And there's the Schaefer-Fulton stain. You have to figure out what's going on with this stain, and then what's the probable diagnosis. Once again, you should tell me the exact disease, specific disease, or the species causing the infection. Okay? It'll be easier if you can just give me the species, because you look in the lab and find the species. All right, any question about any of this? If not, it's 7:20. I'll be here until 8 o'clock to answer any questions on the lab. So go ahead and get working on it. I'm going to stop the recording.